So I'll do the introductions since I know Dr. Celestine Wanjala well. So she's our presenter today, and she is an assistant professor in the Vanderbilt Division of Infectious Diseases, and someone I think that we've all known for quite some time, but with a storied background, which I'll just sort of briefly summarize. So Sally got her initial Bachelor of Science from Cornell University with an emphasis in research, and then traveled to New York City to do research in the Division of Pharmacology, Clinical Pharmacology at Columbia University. She then traveled from there to Thomas Jefferson University in Philadelphia, where she completed a MD as well as a PhD in immunology and microbial pathogenesis. And we were very lucky to recruit her then to Vanderbilt, where she completed her residency as well as her fellowship in infectious diseases. And as part of that, she was a member of the Harrison Society, which is essentially an honor society with an accelerated residency for individuals who are clearly going to have a successful career in research. And after, while she was still a fellow, she then joined the faculty as an instructor as part of the V Scholars K-12 program in HIV and heart, lung, blood, and sleep-related comorbidities. And then when it was time to get a Career Development Award, I think she thought it was supposed to be Career Development Awards, plural. So she's one of the only people that I've ever seen who received a K-23. And within, I think, approximately 3 months of that, also received a Doris Duke Career Development Award, as well as a Burroughs Wellcome Career Development Award. So a trifecta, really all within one summer. And then from there, it's just taken off. So she's received AHA awards, well-scored R01 applications, which I think are still pending, but look very promising, as well as just a variety of different speaking opportunities, engagements, and collaborations. So Dr. Wanjala today is going to be talking about some of her research. We used to say she was a rising star. Now I would probably say a risen star in the field of HIV and cardiovascular disease, and particularly really in the cutting edge field of how do antiviral responses potentially get tilted to go from something that's beneficial to potentially something that's detrimental and can accelerate the development of atherosclerosis and other cardiometabolic diseases. So with that, Sally, I'm going to turn it over to you, and thanks for coming to tell us about your work.
Thank you so much. Thank you to John, the whole CFAR, for this invitation to speak with you a little bit about the research we're doing. So if you don't know, John Carter, Simon Malal, who are part of the CFAR Spiral columns, have been sort of my mentors, and in addition to providing resources, as well as the cohorts, as you'll sort of see when I go through this talk for the next 30 to 40 minutes, really, I stand on the shoulders of giants. I'm trying to interpret that from Swahili, so that might have come out a little bit different. So I'm very grateful to them, and I will highlight a few of the things that I think are interesting. And part of the reason for highlighting this in the CFAR-Wide conference that will be televised is so that folks can start to look for these cell types in the research that they're doing. The only disclosure I have is that I have consulted in the scientific advisory for Gilead, but that does not affect my talk and what I will tell you today. So in the next 45 minutes, I will paint a picture of what it is to live with HIV today. Specifically, we've made a significant progress with antiretroviral therapy. And so other things other than the infection with HIV are coming to the forefront as what our patients are dealing with on a daily basis. And our groups are specifically interested in cardiometabolic diseases. And while my research mainly focuses on cardiovascular disease, I'll look at some of the traditional risk factors that are associated with that when we're looking at inflammation. I will then touch a little bit about inflammation and aging, and beyond looking at systemic markers that are in the plasma, we have the ability to actually look at immune cells. And because we actually have a really sophisticated system that's been set up here with the Immunology Consortium, I think we have the ability to look beyond just one cell type and have made discoveries based on that that I would like to share. And so specifically, I'll paint the picture on this immune complexes as has been pictured here. This is actually a picture we've captured from our cells from the studies that we're doing here. Share data both from the HADM cohort, which is a cohort that was established or recruited by Dr. John Carter, as well as the CINIX cohort, which I think is a wonderful resource for HIV researchers who want to study the human immune cells without necessarily needing to recruit these cohorts on their own by leveraging already established infrastructure in clinics, and then talk about future studies and acknowledgements. So I really like this, even though it's getting a little bit older now, based on when it was initially. I really like this because I think it's a good depiction. I haven't found anything yet that's been published since 2017 that nicely depicts the change that we have in our patient population in terms of what our patients are dying from. So the proportion of annual deaths due to AIDS, as you can see on the left, beginning from 1996, which is where the arrow shows, going down, we've seen a significant decline in patients dying from AIDS. And this is because we've made significant improvements in the antiretroviral therapy. Our patients have choices to the point now I have 3 of my patients who go to clinic and get injectables once a month because they either do not want to be reminded that they have HIV by taking a daily pill, or their work schedule does not allow for it, or they just wanted to change it for other reasons that were found okay by insurance. So we have come a long way and our patients are thriving. But what we've noticed is that they're aging at a faster rate, and we're seeing an increase in cardiovascular disease, cardiometabolic diseases, cancers, all related to accelerated aging that we now know is because of this level of inflammation that our patients have. In this study by Bhatt et al. That was published in AIDS in 2009. This is from the VAX cohort. What I hope you can appreciate is that while they showed that HIV itself is not a risk factor for diabetes, HIV infection amplifies the traditional risk factors for diabetes. So if you go from the age category of less than 35 at the top, all the way to 65 to 69 at the bottom, what you can appreciate from people without HIV, which is abbreviated as PLWH on the right, is that as you get older, there's an increased risk of diabetes. However, if you have HIV, by the time you're 45 to 49, your risk is almost as much as when you're 65 to 69, and it's even much more as you get older. So there's a steep increase, and one of the factors that this also interacts with is with BMI. And so we know that with that there's an interaction between traditional risk factors and um, and HIV in terms of increasing the risk for diseases of aging, such as diabetes. In this pooled study that was done, or a meta-analysis by Shah et al. In 2018 that was published in Circulation, again, I think we nicely depict here that the risk of cardiovascular disease in people living with HIV referenced against people without HIV is significantly higher. And for us, and for those of us in the field, we'll usually state a number of twofold higher, and it mostly comes from this study. And other studies have gone on to show about the same ratio. So looking at cardiovascular events, or if you break it down by myocardial infarction and stroke, if one has HIV, their risk of cardiovascular disease is twofold higher. And while there are other studies that I'm not going to mention, there's enough evidence that suggests that our patients have increased risk of developing cardiometabolic disease, increased incidence and prevalence, even when you account for traditional risk factors, even when our patients are on antiretroviral therapy. And so why is this? And one of the reasons, and I think the main reason, which is also appreciated in people without HIV, is that inflammation plays a significant role in aging. But if you have HIV and are on antiretroviral therapy and are undetectable, as you can sort of see here, you still have elevated biomarkers. And these are biomarkers that can be measured in the plasma, but they're still very important in terms of the risk for cardiovascular disease. And so this is unadjusted when you adjust for age and gender, and also for race. Among those who are undetectable, and here they sort of counted as viral loads less than 400, HSCRP was 40% higher in people living with HIV. IL-6 was 60% higher, and D-dimer was 49% higher compared to the MESA multi-ethnic study of atherosclerosis. So these are folks who also have a high risk of developing atherosclerosis. They're otherwise healthy without HIV compared to folks from the SMART study. So we appreciate the fact that our patients living with HIV on antiretroviral therapy who are doing great to remain undetected with undetected virus have a high level of systemic inflammation. Now, most studies from the past have mainly looked at markers that can be measured in the plasma. This was cheaper, it was easier to do, but we now have the ability of actually looking at what immune cells are present and understanding this more. And I like this picture that's depicted from a review in endocrinology in 2018. Inflammation is the center of aging in many ways, and it alters not only the epigenetics and sort of this imprinting that unfortunately can survive for a very long time, even with change in behavior, but also in the ability to sort of regenerate when you think about stem cells, metabolism, and how well our cells are able to uh, process or are able to metabolize whatever we're ingesting, and then the way that they because of how the metabolism happens, you can either have a pro-inflammatory or an anti-inflammatory phenotype, and then as well as how it interacts with stress, proteostasis, or macromolecular damage. So inflammation is in the center of a lot, and on the very outside of that is aging and aging-related diseases, of which heart disease is the #1 killer globally, and then diabetes is a risk factor for heart disease. So what are the different immune cells that we think about? And this cartoon simplifies it, but I think it's sort of a good way to think about it so that it's not overwhelming. But we have our antigen-presenting cells that are dendritic cells, monocytes. B cells at the bottom here make antibodies, but they can also present antigen, whether appropriately or aberrantly. And then macrophages, you don't find these in circulation. They're usually in tissues. And monocytes beget macrophages. Monocytes can also beget dendritic cells. And then you have T cells that most people are familiar with. There's CD4 cells that tend to be thought about as helper cells, but they can also fight infections in other cells. And CD8 T cells tend to be the cells that you think about will go and sort of blow up cells that are infected. And they have tools such as a mechanism such as perforin that are able to make holes in cells that are infected and actually cause the cells to die, and that's how they fight infections. So while I'm simplifying it, it's quite a complex system where these cells are able to interact either by touching each other, or they're able to interact or inform the rest of the body what's happening by secreting cytokines such as IL-6, interferon gamma, and through this sort of sophisticated, really intricate system, they're able to prevent us from getting infections, but at the same time, they're also the same drivers of inflammation that can drive like, for example, diabetes and atherosclerosis, depending on our diets, our genetics, so many factors go into that. On the right here at the bottom, I highlight a monocyte and a T cell interacting, and with some of the molecules, MHC molecules or T cell receptor that allows them to sort of talk to each other and inform each other about whether there's a pathogen or if it's a vaccine, making a good vaccine response. And so what most of us might be familiar with is on the bottom right here, the CBC. So when we get labs from our patients, we sort of know that if the white count is really high, they might be infected. If it's really low, it's not a good sign. They might have cancer or AIDS, HIV. At the very bottom where you see the differential, we're able to sort of look at neutrophils, lymphocytes, monocytes. This is sort of more information that we're able to get from a CBC and diff. Now, in clinic, you don't think about it as much as we do, but in the lab, we're able to even go further and ask more specific questions about the lymphocytes, which are sort of these cells, the lymphocytes, the monocytes. We're able to characterize them a little bit more to understand how are they behaving, what are they doing, are they maladapted, or are they actually promoting a good response that can help fight HIV, for example, in elite controllers. In 2019, there's a team from California led by Burrell et al., who were interested in studying monocytes. And what they did is that they sorted this monocyte population of cells by flow cytometry, and then they ran RNA, they sequenced the cells in bulk. And what they noticed is that these cells looked like they were also T cells. So initially they thought maybe they had discovered a new cell type. But then somebody astute said, you should go back and make sure that it's not a new cell type that both expresses CD3 is a marker of T cells, it's only expressed on T cells, and CD14 is a marker of monocytes that's only expressed on monocytes. And so when they went back at the institution, they marked the monocytes red based on CD14 expression. They marked the T cells green. Can you see my marker? Is that better? Okay, and then they were able to at least tell the monocytes and the T cells. And what they discovered is that there was actually a group of cells that were both CD3 positive, CD14 positive. And when they looked in the imaging flow cytometry, they were able to see that it was actually two cells, or in this case, even three, that were sort of attached together and they were sorting together, even though their goal was to sort the CD14 cells. And so when they sequenced them, they got genes that were for both the T cell and monocyte. Now, there's a small group of cells here where it's only either the T cell or the monocyte or both. And in some cases, the bond, or when these cells are sort of interacting and sharing information, sometimes when they separate, they can take a membrane of the other cell. It's something that's called trogocytosis. You don't have to memorize it. But that could be another reason why you might find a cell that's expressing both colors. So the reviewers, and after talking to Burrell, I had a chance of meeting her in AAI recently, which was super exciting. But the reviewers sort of thought, well, when you take blood from patients and you process them and you put them in the freezer, it's very likely that you're going to see some of these cells stacked together just because it's an artifact of cryopreservation. So what they did is they recruited patients, they got fresh blood, and then they also, from the same exact sample, quickly cryopreserved it in liquid nitrogen and then thawed it, and showed that there was a strong correlation between these complexes being present in the frozen sample and the fresh blood, so that this was really not an artifact. And they went on to characterize the synapse, which is the area where these cells talk to each other, as you can appreciate here. And they found that you actually have these molecules, when they're forming a connection, they tend to recruit a lot of them there so they can form a strong bond. And in that way, they can have good communication of what the antigen is, that the T cell tells, that the monocyte tells to the T cell. Then the T cell gets activated, and it gets activated to the point where it can actually then go fight the infection effectively. So they're able to show that those are the molecules that should be recruited to the synapse that I'm highlighting here, and they've highlighted in the box, were actually being recruited, and that this was forming a relationship that was important. But even more important is they looked at three different settings. They looked at a cohort where they had folks who had had active tuberculosis, and 2 months later, they had already had treatment and with improvement in clinically, and they found that these complexes that they found at diagnosis were reduced significantly in all the participants. And then they also had a cohort of people who had been infected with dengue. Now, if you're infected with dengue and it's a severe form of dengue, then you can have dengue hemorrhagic fever. Sometimes that happens when you've seen different serotypes of dengue. And they showed that compared to those who were previously infected in the black, that those who had dengue fever, that was not significantly different, but those who had dengue hemorrhagic fever had significantly more of these complexes. And finally, in a setting where you vaccinate patients, if patients are healthy, they're not sick, and you vaccinate them, and 3 days later, the immune system should be ramping up. So this is when whatever you vaccinated with in the tetanus vaccine is being presented by the monocytes to the T cells so that they can set up the immune response that will then become memory and prevent these people from getting tetanus in the future. You can see that there's an increase in this monocytes. So whereas historically we've always ignored these artifacts, these complexes, as though they were artifacts, Burrell et al. Were one of the first to pay attention to these enough and show that they actually seem to be functional and they're going up and down as you might expect after infection or after vaccination or after improving from a vaccination. So why am I telling you about this? Well, a few years ago, I'm not going to age John, when he got his first R01, he established the Hatton cohort. And one of the interest, he was interested in sort of diabetes and inflammation, looking at both the adipose tissue and the blood. And we've presented those results in the past, so I don't talk about them today, and we've published them. But then one of the things we wanted to sort of understand, especially as systems were getting a little bit more complex, is if we use a multi-parameter system where we can actually look at all of the different cell types. And so we used mass cytometry, or we call it CyTOF. And early on, this was one of the only ways that we could do that. The question we asked is that what are the cell types that are different by diabetes? And can we find these cell types? So that was one of the main questions. And one of our outcomes, in addition to diabetes, which is what this cohort was established for, was cardiovascular disease, because we were calling these patients back to get CT angiograms on them. And I think historically, even from this cohort, we had only used panels where we could only look at T cells or just monocytes. But here we had the ability to look at all the cell types together. So data that we might have missed, we actually were able to capture. And a lot of this analysis was done by a talented scientist here, Josh Simmons, who does a lot of our flow cytometry analysis and is also doing a lot in R. And so what we found is that when we looked at patients who did not have diabetes, there were 14 of them, and then we combined those with prediabetes and diabetes. This was during COVID, so we were sort of limited with how many patients we could call back. And so there were 28 patients in the prediabetes diabetes group, so we combined them. What we noticed is that there was this cluster of cells that were missing in the folks without diabetes that were definitely present in folks with diabetes. And while they were not uniformly, while not each patient uniformly sort of contributed to each of these clusters, it was definitely coming from one group, which was those folks with insulin intolerance. And on further investigation, we found that these cells were actually complexes. Now, with the initial, we were not able to do what Burrell did, because with CyTOF, when you ran your samples through that machine, the cells actually, after they pulverize, it's part of the way that you're able to detect what markers are bound to them. So we went back and stained the cells just very simply with CD14. So this is the turquoise that you see here. And so just to orient folks who are not used to seeing these types of two-dimensional plots, the FSCA, the x-axis here shows you the size of the cell, and then the x-axis, the y-axis is the granularity. So monocytes are bigger, and they tend to have more, they tend to have more like the vacuoles with like perforin and not perforin, but granzyme. So they tend to be more granular. And then T cells. So this bottom, at the bottom here is where we'd expect to see T cells, which are in green. But what we did notice is that you had T cells that were showing up where the monocytes should show up. And then indeed, and these cells are not dead. So sometimes if cells are dead, you're going to see them light up in all the different different gates that you look at. But here we were able to show that if you looked for the cells that were live dead, the dead cells tend to sort of be in this bottom corner or here, that these cells were indeed alive. And when we gated on the CD3 live cells, we were able to find that we could detect cells that were both CD8 positive and CD14 positive here after gating on the CD3 cells. And then and then some, I guess here they show the same gate. And then here I'm showing you that, in terms of the marker for CD14 and then also the marker for CD8. So there's CD8 positive, CD14 positive cells here that are also, that go along with what Burrell had shown. But, pictures is worth 1,000 words. So we did sort these cells, and we also sorted CD14 cells or just the T cells on their own. And I hope you can appreciate that there are cells that are there are more cells that seem tightly in friendship or clustered on the right compared to the left, where there might be a few that look like they're touching, but they're really not. But I think John said we still have to show more, and Simon said we still have to show more after this. And so we did a time-lapse imaging. And I hope you can appreciate that after letting these cells sit and imaging them with time-lapse imaging overnight, but here we only capture about 250 minutes, is that you have your big cell here, that's the monocyte. You have the T cells here. And we even captured on the top here a T cell that was dividing. And we even captured cells where you have these monocytes that are going and sort of checking out the T cells, but they don't last there long enough to sort of form a pair and form a bond. And yeah, that sort of Dr. Mandrink helped us sort of make those videos. So we sort of at this point had shown that, you know, while we were not expecting it, we had found that people with HIV and diabetes, which nobody had shown before, seem to have more of these complexes, and that these complexes are they seemed jet true because when we image them, we can actually find them. And when we put them overnight, that some of them remained in complex, whereas some of them actually left the complex, which, potentially is expected. Now, we wanted to see whether or not there was a correlation or an association between these clusters here that are boxed. Those are all the complexes, and hemoglobin A1C or fasting blood glucose, and whether or not it was the CD4 T cell count. We looked at other things, but we found that there was a positive correlation when we adjusted for age, sex, BMI, and LDL between the proportion of these clusters and mostly hemoglobin A1C. Fasting blood glucose, there was a trend. However, it was not as significant. And this is probably because fasting blood glucose is more likely to be affected, by what somebody ate, or as compared to the hemoglobin A1C, which is, even in our patients with HIV, is probably more telling us how they have been for the last 3 months, even though we know that other factors such as anemia, other things can affect it in our patients with HIV. Um, and then furthermore, we went on to characterize these cells a little bit more. And I think one of the questions that had come up in the Burrell paper, which we addressed here, this is what this is addressing, is whether or not you're just going to see more cells complex just because of the proportion of T cells and monocytes. So this is taking the proportion of T cells and monocytes into account. And even when you adjust for that, you continue to see that you have more of the CD4 complexes with monocytes and more of the CD8 complexes with monocytes as well. And it's higher in people with diabetes. In a longitudinal setup, sorry, that's my phone. Um, so in when we looked at it in longitudinal samples, we found that these actually sort of remained. So if you remember what I showed you with the Burrell paper, if you have an infection and it gets better, they should go down. If you've been vaccinated, they go up, but you think that they will go down because that antigen or whatever was driving that is not there anymore. But what I hope you can appreciate is that there was an increase in both people without diabetes and with diabetes. And there's variation. In some people, it actually goes down. So there's enough variation between two time points from the same individuals. However, there's a general trend to them either staying the same or even increasing in some individuals. Now, we did not have enough of a time lapse, and this was during COVID when we had the 2 visits to get a signal for whether those who had an increase in this and those without diabetes actually went on to have diabetes. That was one of the questions that has come up in the past, and it's something that we definitely think is worth exploring in the future, in future studies where we have longitudinal samples. And here, for those who sort of double in this a lot, I think a majority of the cells are CD8 cells that are in complex, but you have CD4 T cells, and they're mostly memory cell types. So there's central memory and effector memory. There's not as many naive cells, which is not surprising because we think that these cells, by the time we're finding them, are being primed, and so they should be effector memory cells or central memory cells. Not as many TEMRA cells as well, but there are a few that were there. And then because, we were studying HIV, one of the questions that we had was, could we potentially detect HIV in these complexes? And so we leveraged droplet digital PCR. This was done by Rama Gangula and in Simon Malal's lab. And so what we did is we used the LTR primers and we asked, can we detect copies of HIV? So the blue is the LTR primers that I used to detect HIV. And the good thing about this method is you can detect up to 1 copy of HIV. And then the green dots are RNase-P. There's 2 copies of RNase-P per cell. So it allows us to be able to quantify how many copies of HIV we have. And what was clear is that if we looked at the viral copies of HIV per million cells, it actually looked like we were able to detect more HIV in the complexes than we were detecting in just the CD4 T cells. So is this that you have CD8 T cells that are sort of going after monocytes that are infected by HIV? That's potentially a thing. And so I think more needs to be investigated. And now that we sort of can potentially have antibodies or even RNA-scope probes that can be used in these assays, I think that that question can be answered. Again, when you look at the CD4 T helper subsets, Th17 subset was significantly higher in folks with diabetes compared with without diabetes. All the cell types seem to be going up. We also have sorted these cells and looked for their T cell receptors, and they're oligoclonal, but not just one clone. But anytime you have more than 2 of the same TCR, especially there's not that many of these cells, it points towards that there's potentially a specific antigen that's driving this, and some of these are HIV specific, some of these are CMV specific. Based on what's been published in the past, we have not interrogated these specifically for what they are. I showed this to somebody and they were not sure what they are, but this is a TCR, it is really tiny, but it's a TCR alpha beta pair. So with this information, we can actually put these T cells into reporter cells and be able to interrogate what antigen they're binding to. Okay. And then the other thing that we've done is transcriptionally, we're only limited by, so when we look at this by flow cytometry, we're limited by the markers we put in. When we looked at them transcriptionally, we sorted the complexes and did single cell sequencing. And then we just took T cells and classical monocytes that were also sequenced and combined them in the same ratio. And we found that those significantly more granulysin, and then also HLA, class 2 HLA in these complexes, and then also some mitochondrial genes. Moreover, we were able to sort of look and interrogate what energy potentially might be keeping these cells active. So this is using an assay called SNITH. I'm not going into details, but what I can tell you is that if you look, the complexes are here on the first, that these complexes rely on glucose for energy, and compared to monocytes on their own, they also rely on mitochondrial, so oxidative phosphorylation. And this is more than naive cells on their own. Now, somebody could say maybe it's because they're paired with T cells that this is higher than the monocytes, and it's possible. Now, what we asked is that, do you see a difference in whether or not you have these complexes if you just put the cells in puromycin, which is sort of how we know what energy system, so this can be looked at as the control. But if you block glycolysis with 2DG, do you see a difference in the number of complexes? And the answer was no, not significant. But if you block it with oligomycin, and this was just for 30 minutes, you see a significant decrease in the complexes. So it's possible that oxidative phosphorylation is one of the main ways that's maintaining these complexes. And so just sort of a whirlwind here, just to sort of talk about the CNIX cohort, because this is what we propose to use for our for the R1 that is maybe being funded. We got samples from the CNIX cohort for proof of principle to see whether This is not just from the HADIM cohort, that we'll also see it in the CINIX cohort. So a lot of the samples came from UAB, some came from Case Western. And so we had folks who had diabetes with an A1C greater than 9, less than 7, no diabetes, and pre-diabetes. So there's a total of 62 folks. They were well matched for age, sex, and race. In terms of viral copies, here it looks like it's significant, but they're all undetectable. It's just based on the lab differences. The CD4 T cell enrollment was different, and then not surprising, they were different by diabetes. What's different here is we now use Cytec and not CyTOF. So going forward, we'll be able to potentially sort these cells and study them outside of looking at them by imaging. And just to sort of show you again how we gated on this cell, so you have the monocytes there, and then, oh, I didn't show the rest of it, but we gated them the same way that I showed you in the other paper, where we had CD3 positive, CD14 positive T cells that were complexes. And what we did different for this study is that in addition to just looking at them when they were in media, rested overnight, we also stimulated the cells with PP65, and then also this is TB, inactivated TB, whole TB, because one of the questions we wanted to see is that if you take these cells and activate them ex vivo with an antigen that they've likely seen, which is PP65, for TB, our patients are mostly from America, so we didn't have many who were TB positive. But in terms of activating the monocytes, we wanted to see what that would do. And then what I hope you can appreciate here is that starting on the left, if you compare to media only, we saw an increase in activated monocytes. These monocytes are expressing both interferon gamma, TNF alpha. They're CD14 positive. They're classical monocytes. But then if you look at, and then at the bottom here, you see with none of these patients had TB, so you don't see activated T cells that are TB specific. However, you see, well, maybe there's a few here based on this p-value, but you see significantly more cells that are activated by PP65, but you don't see an increase in the complexes. And Burrell showed this as well. So just putting the cells ex vivo with peptides does not lead to an increase in the number of complexes. If anything, there's probably a decrease here compared to media only. And then the next slide just sort of shows the paired samples. Refusing to move. Okay, there we go. Okay, and then when you do the paired analysis, again, same thing. This is pairing the sample from the exact same patient. The trend is about the same. There might be a few here that went up, but nothing that's significant to suggest that ex vivo, just putting these cells with an antigen will come up. Because this is one thing that came up with our previous publication, is we sort of are trying to figure out a way where we can ex vivo drive the formation of these complexes, because it allows us to study why they're there and potentially why they're persisting in people with HIV with diabetes compared to other folks without diabetes, and then harness it if it's a good thing. So for example, for vaccines, that actually might be a great thing for folks who are older than 65 so they can have a good response. But for chronic infections or chronic disease where it's driving diabetes and atherosclerosis, then we definitely don't want that. But I think what was reassuring is that we found same thing that we found in the HATIM cohort, is that when you look at them based on diabetes, that folks without diabetes have significantly fewer of this. Now, if you have good diabetes control, that's potentially a little bit decreased, and even bad diabetes control, suggesting that there's some medications that our patients are on when we start them treatment for diabetes that brings them down, but does not quite bring them to the level of that without diabetes. But I think that's definitely modulating it to some extent, but no difference otherwise between all the three groups. The trend is about the same. So I've said a lot, but the main things that we have sort of found is that in people living with HIV, we know that they're living longer, we're achieving aviremia, but they still have a high risk of diabetes and heart disease. And while we know chronic inflammation, which we've mostly measured by systemic inflammatory markers, is important, we think that with these complexes that we're finding to be higher in folks with diabetes or diabetes and risk for cardiovascular disease, we're also going to recruit patients with cancer who do not have diabetes. We think that there might be information that we can learn from some of these long-lasting interactions for what might be aberrant, but actually what might also be helpful, especially if we can figure out a way to make these happen ex vivo. And so this is just summarizing that. And what we know from our study and also Burrell, that just giving the cells the antigen that might be specific for that specific T cell doesn't seem to have us in a span of 6 hours capture those complexes that remain in complex together. We do need to do this also with HIV negative samples at the same time, which I think to see what the role of the reservoir is, because we know we can detect the reservoir in this cell. So ideally having a longitudinal sample where we're planning to do this over 5 different time points with a lot of metadata and understanding what's keeping them together will provide a lot of information. And like I said, it takes a village, and this is just like a fraction of the folks who have helped get this research done. So I definitely want to share my gratitude to all of them. And even more importantly, it's our patients, the CCC, through the cynics cohort who have done a lot for this. And with that, I will take any questions. Thank you.